Okay, let's talk about chemical digestion a little more. Now, what is chemical digestion? Again, it's going to take the food that you help to break apart with mechanical digestion, and now you're going to use chemicals and enzymes and other things to help break it apart into smaller and smaller molecules. Now, what are enzymes? Well, again, most enzymes are proteins, and in terms of digestive enzymes, they are pretty much mostly proteins. And what they're going to do is break down biological macromolecules. So macro meaning big, so they're big molecules that were broken apart during the process of mechanical and chemical digestion, but now you want to break them into smaller and smaller molecules. And acids, so what acids do is, or some of the things that acids do to help with digestion is to denature proteins. So proteins are folded, and if you go all the way back to cell biology, when we talked about primary, secondary, tertiary, and quaternary structures, so basically you get that chain of amino acids and you start folding and folding it until you get 3D structure. Now, denaturing by exposing it to a lot of the acids or exposing it to a high acid concentration is going to disrupt some of the bonds that hold the proteins together in that 3D shape, and now it's going to unravel it into a linear form again. Now, you also have like some enzymes, they actually operate best at some acidic pHs, some operate better at a neutral pH, but also acids also activate some enzymes as well, such as pepsinogen, when we talk about the stomach, gets activated by hydrochloric acid to become its active form, pepsin. But we'll save that for when we talk more about the stomach. Now, what are our biological macromolecules? Well, our biological macromolecules go back to all the way when we talked about chemistry and the cells in the AMP1, but carbohydrates, that's one of them, or also known as sugars, and protein, and then we have our fats and lipids, and then fourth type of biological macromolecule are nucleic acids. And where can you find 3 out of 4 of these? You can find them on your nutrition labels, and this is in the US, so if you're an international student, then this might be unfamiliar, but if you just go to the United States and look at this is on the back of everything that you can, every food product. Now you can find 3 of these, and you can find your carbohydrates and the sugars over here. You can find the protein over here and the fats over here. Now, why can't you find nucleic acids? Well, nucleic acids, all of these have calories, the first 3 over here, but nucleic acids, unless it's very unusual, starvation, physiological conditions, you're probably not going to use nucleic acids for energy. Okay, so why are macromolecules so complex? Well, a lot of the macromolecules are made from smaller units, smaller molecules, and then they're built up to make bigger and bigger molecules that are more complex, hence macromolecules, big molecules. Now, a polymer is a chain of molecules with a repeating structure, and that repeating part is important for a chemical or macromolecule to be a polymer. So here we have, say this is a little nutrient over here, and then what if we had a polymer of this nutrient? Well, again, it has to be a chain with a repeating structure, and it can have branches as well. But if we have a chain of these nutrients, now we have a polymer of this nutrients. Now, carbohydrates, amino acids, and nucleic acids, they can form polymers. So what we see here is that we have this little nutrient, and now it extended and repeats and it lengthens. In theory, polymers can extend until infinity, and so can these long chains of carbohydrates, amino acids, and nucleic acids. Of course, our bodies are finite, so they're not infinite, but they can form these chains of molecules. Now, fatty acids and lipids, they do not form these polymers, and I'll explain why shortly. Okay, now let's talk about carbohydrates. So here's a single carbohydrate by itself, and when you have one carbohydrate by itself, it's a monosaccharide. So mono is that prefix like a monorail or monocle. That means one. Then when you have 2 monosaccharides linked together, what do we have? We have 2 carbohydrates, so now it's a disaccharide. So this disaccharide, what if you keep on adding single monosaccharides and have a polymer of carbohydrates? So if you keep on daisy chaining and having these, adding more and more carbohydrates and lengthening this chain, eventually you have many carbohydrates, and that's what we call a polysaccharide. So again, poly, that prefix meaning many. So many polysaccharide, just many carbohydrates linked together. Okay, so let's talk about examples of these carbohydrate macromolecules. Here we have a long chain of carbohydrates linked together, but notice this chain is unbranched, meaning that it's just linear and it's repeating over and over and over. This is a general type that we call amylose, and amylose knows that, again, there's no branches, but what if you start having branches to this structure? Well, one example of this are starches you find in things like plants. So plant starch is a branch chain or branch type of carbohydrate macromolecule, and that's what we have here. And glycogen is actually another form of carbohydrate macromolecules we have in our own body. So what's the difference between starch and glycogen? Well, this is in general, glycogen tends to have more branches than starch. So yeah, here we had starch was branched, but glycogen seems to have more of these branches overall compared to a starch molecule. But that's not a very exact definition, but that's just more in general. Alright, so we talked about carbohydrates. Let's now talk about proteins. Now, there are 20 asterisk different amino acids. And what I mean, like, why is there asterisk? Well, there is that 21st amino acid, selenocysteine. Everybody has selenocysteine in their body, but that's more if you're going into cell biology. You might want to learn more about that. Or actually, selenocysteine is important, but in terms of like the basic amino acids that we use to fold most of our proteins, 20 canonical ones. And we're not talking about the 22nd amino acid. I think that's only found in like archaea bacteria and something like weird like pyrrolysine. And it's like, don't worry about that for this class. This is human anatomy and physiology. But basically what I'm getting at here is that not all amino acids are the same. But what if you take a bunch of amino acids and start to daisy chain them together and link them together in these things? And the classic ones type of bond we use to link amino acids in our cells to make a protein are peptide bonds. So when you have peptide bonds and you're making a protein, you're going to take a bunch of amino acids, make these peptide bonds between them, and then you'll have many peptide bonds. So remember what we call when we have many monosaccharides linked together? We call it polysaccharide, right? Now we have many peptide bonds, so we're going to call this a polypeptide. So it's a long, unbroken chain of amino acids. Now that's what we have with this. So again, this is a polymer, and yes, a polypeptide is a polymer of amino acids. Okay, let's talk about nucleic acids real quickly. So if you want more detail, go to the cell biology part of our course. But what we have here are different letters of these nucleobases of our nucleic acids, and these are the ones found in DNA. So we have thymine, adenine, cytosine, guanine. But in RNA, instead of thymine, you have uracil. Now, how are these layers arranged on these nucleic acids? Well, again, nucleic acids are a type of polymer. So what you can take is a bunch of these nucleotides and link them together. And this is how we get the sequence of all these nucleobases we see here. So I like to do a little analogy that this is kind of like a little friendship bracelet or a message bracelet. So with these like bracelets, you have these repeated beads or links that link together, and you may have little letters on them. Nucleic acids are somewhat similar. They have a constant backbone right here, so this is like the chain part right here. This sugar phosphate backbone runs throughout the entire nucleic acid, but it's the letters that dangle off of it that differ. And in this case, the letters have information, so it spells out a word. Same with nucleic acids. The sequence of these letters, these nucleobases, they tell the cell what type of amino acids are associated with these sequences and other things as well to determine the structure of nucleic acids. A lot of information comes from this code of nucleotides. Now, if you want more information on this, you can go back to my cell biology lectures from the previous semester. Now, let's talk about fats and lipids. So fats and lipids are macromolecules. That is true. So fatty acids, triglycerides, cholesterol, these are all examples of fats and lipids. But do they form polymers like carbohydrates, like amino acids, and nucleic acid? So one example of these macromolecules, well, here we have something called glycerol. So glycerol isn't a fatty acid or a lipid by itself, but it is important for the storage and release of fatty acids. So fatty acids are a group of compounds in your body, and there's many types of fatty acids. We'll talk more about them when you talk about metabolism. But how does the glycerol relate to these fatty acids? Well, glycerol is able to molecularly bind and combine with these fatty acids. And if you have three fatty acids and chemically bond them to a glycerol molecule, you'll end up with something we call a triglyceride. So triglyceride is, and this is a common sticking point that people think, oh, it means 3-glycerol. No, tri in this case refers to the three fatty acids that's bound to a single glycerol molecule. But with this triglyceride, what do we notice? Well, there are 3 oxygen atoms on the glycerol molecule, and these are the anchoring points for these fatty acids. So when you have all 3 of those oxygens occupied by fatty acids, there's no more sites for another fatty acid to bind to this glycerol molecule. So we have 3 fatty acids bound to glycerol, and that's it. We don't have any more oxygen atoms left on glycerol, therefore this cannot keep on extending until infinity and repeating like a polymer. If fatty acids could form a polymer, then it would have to look like something like this, where it can continually repeat, and form a long chain. This does not happen in physiological conditions. So fatty acids by themselves do not form polymers in your body. Now, we talked about another type of lipid, cholesterol. But does cholesterol form chains, or does it branch like this? No, it doesn't. So cholesterol doesn't form polymers in your body either. So the lipids in your body do not form polymers in physiological conditions because they can't form these repeated, branched, or linear structures over and over and over again. In terms of like glycerol and fatty acids, we just have only 3 fatty acids max, and that's it. Now let's talk about macromolecule enzymes. Okay, so the first type of macromolecule enzymes we're going to talk about are carbohydrates. And if it's taken A and P1 with me, you might know these 3 letters. If you have a suffix and it ends with ASE, it probably is an enzyme. So what do carbohydrates do? They're enzyme for what? Well, they're going to catabolize, and that catabolism means to break down something. So they're going to break down complex carbohydrates like amylose, starches, and glycogen we talked about earlier. Now proteases, what are they going to do? Well, that sounds pretty obvious, right? They're going to catabolize and break down proteins in polypeptides into smaller peptides and amino acids. Lipases, what are they going to do? Well, lipase sounds like lipids, right? So they're going to take triglycerides that we talked about earlier and break them down into glycerol and fatty acids. And then nucleases, what are they going to do? Sounds like nucleic acids, right? So nucleases are going to break down nucleic acids into smaller nucleotides. Okay, let's fast forward a bit. So we will talk about metabolism and how you're able to actually convert these molecules into energy. But let's skip to the punchline, or preview the punchline. So say the macromolecules are broken down, your body absorbed them. Now you're trying to convert these molecules into energy. So what is the payoff from converting it into energy? Carbohydrates over here, well, when you convert them into energy, how much energy do you get? You get 4 kilocalories per gram. And why do I specify that? Now, kilocalories is equal to 1,000 calories. And just to play it safe and emphasize it even more. So whenever you see these calories, notice that they're uppercase C. So uppercase C is a kilocalorie, versus a lowercase c would be actually 1,000th. So 1 over 1,000 of a regular calorie. So when we see these values here on a nutrition label, they're talking about kilocalories, but I'll use this notation just to emphasize and be very, very clear. We're talking about kilocalories, not just small lowercase calories. Now proteins, and do I want you to memorize this, like the values like to the 100th decimal place, because I know they have more specific values, but let's just keep it simple, rounded to the nearest kilocalorie. For proteins, what you have is roughly the same as carbohydrates. So 4 kilocalories per gram, that's the value we'll use for this class. Now fatty acids, these are 9 kilocalories per gram, so weight by weight. So if you have a gram of each of these, fatty acids are going to have more than twice the energy content than carbohydrates and proteins. Now fatty acids have energy, but look at cholesterol and notice that it's a different line item. So cholesterol is a lipid, but it's hard to actually break down cholesterol into smaller molecular units your body needs to convert to energy. So actually cholesterol, even though it's a lipid, the energy yield from cholesterol is negligible. Your body needs a lot of enzyme and effort to actually convert cholesterol to a form that's usable by your mitochondria in your cells. Nucleic acids, again, for this class, we're not talking about those strange like starvation conditions where your body would have to actually scavenge nucleic acids and convert them to energy. So for this class, this is another reason why you don't see nucleic acids on a nutrition label, is because that in people who have normal caloric intake, the energy you get from nucleic acids is negligible. So you might be wondering, okay, so where does this chemical digestion occur and where are these enzymes? Well, they're all over your digestive tract, and the thing is that different parts of your digestive tract, they might have some enzymes, in a different set of enzymes in your mouth, a different set of enzymes in your stomach, and a different set of enzymes in your intestines. So the different organs and tissues of your body, they specialize in different types of enzymes and breaking down different types of biological macromolecules. So that's what we're getting at here, is that you need all the organs to, and they all work together, and no one organ takes on the burden and does chemical digestion for the entire system. They all contribute to chemical digestion eventually during the journey that food takes throughout your digestive tract. And here are learning goals and key points. So your first goal is to list the main type of biological macromolecules that provide energy, including carbohydrates, proteins, lipids, and nucleic acids. And you also have to explain how they are made from these smaller units, these building blocks, and how do you get those big complex macromolecules. And then you also want to list the categories of enzymes. So we talked about 4 types of biological macromolecules. Hint, hint. What do you call the enzymes in general that break down these types of macromolecules? And you also need to describe the energy yield. So how much calories per gram of each of these macromolecules do you get? So from carbohydrates, fats, and protein. And do nucleic acids have energy? Go back to the previous slides if you already forgot.